A tiny finger-painting, milk-spilling toddler is literally more densely wired than a fully grown adult.
Yeah, it's pretty wild to think about.
It really is. So welcome to this deep dive. Today, we are taking a stack of anatomical and physiological sources and going on a mission to answer just one fundamental question. How do neurons actually talk to each other?
Hmm.
We're going to look at how we start with this massive, overwired toddler brain and how it eventually specializes into the adult brain you have right now.
It is a stunning realization, honestly, because, we tend to think of human development and learning as simply adding more and more neural connections as we gather knowledge.
Right, like building a bigger hard drive.
Exactly. But the biological reality is far more dynamic. To understand why that toddler has more connections and how we end up losing nearly half of them just to become functioning adults, we really have to introduce the star of today's deep dive, which is the synapse.
The synapse. I mean, it sounds like the title of a sci-fi thriller.
It really does. But today, we're going to explore this invisible gap between your cells and the incredibly intricate biological machinery that crosses it. We'll look at the fierce, almost petty historical debates over whether this gap even existed.
Oh, the drama's amazing.
Oh, yeah. And we'll see why this tiny junction is the secret behind everything from how your memory works to how you learn a new language to the physical mechanics of why your morning cup of coffee keeps you awake.
Okay, let's unpack this. Let's start with the history first, because before anyone understood how synapses worked, scientists basically threw academic punches over whether these things were even real.
The drama really is fantastic. So, to set the stage, a chemical synapse isn't a physical connection between two nerve cells. It is a gap.
Just a totally empty space.
Right, a microscopic space between two cells called the synaptic cleft. And when we say microscopic, we are talking about a canyon that is typically 20 to 40 nanometers wide. Yeah, that is roughly a millionth the width of a human hair. The cells never actually touch. But if we rewind to the late 19th century, the dominant scientific view was that the nervous system was just one continuous, fused web of tissue. They called it the reticulum.
Like a seamless net where everything is just physically melted together.
Yeah, as the consensus, yeah. And it's championed by Camillo Golgi, who was an Italian physician. Golgi actually invented a revolutionary silver staining technique because before this, brain tissue just looked like a gray mush under a microscope.
You couldn't see any detail at all.
Exactly. But Golgi's stain randomly darkened about 1% of the neurons in a sample, rendering them completely black against a yellow background. And that finally let scientists see the intricate branching structures of nerve cells.
But then a Spanish anatomist named Santiago Ramon y Cajal enters the picture, uses Golgi's own stain against him, and completely upends the reticulum theory.
Right. Cajal takes Golgi's method, spends just countless hours peering through his microscope at embryonic brain tissue, and realizes the reticulum theory is completely wrong.
He actually sees the gaps.
Well, he sees that these are discrete, separate cells. Their branches come incredibly close to one another, forming dense networks, but they do not physically fuse. This revelation became known as the neuron doctrine.
I imagine Golgi did not take kindly to someone using his own invention to dismantle his life's work.
Oh, it was a bitterly contentious feud. And it all culminated in 1906 when Golgi and Cajal were jointly awarded the Nobel Prize in Physiology or Medicine.
Oh, that must have been an awkward ceremony.
It was. Golgi actually used his Nobel lecture to publicly attack Cajal's theory. He stood there on the biggest stage in science, accepting a joint award, and just trashed the neuron doctrine.
The audacity of doing that during a shared Nobel speech is wild, especially considering history and modern biology would prove Cajal was entirely right. But what stands out in our sources is that neither of these guys actually came up with the word synapse.
Right, yeah. Back in 1897, a British neurophysiologist named Charles Sherrington was writing a textbook chapter, and he was studying reflexes.
Like when the doctor taps your knee.
Exactly, or like how pulling your foot away from something sharp takes a fraction of a second longer than a continuous electrical wire should take. He deduced there had to be a gap slowing the signal down.
Ah, I see.
So he believed Cajal was right, and he needed a word for this functional junction. Unhappy with his own ideas, he literally walked over to his Cambridge colleague, a classical scholar named Arthur Verrall. And Verrall suggested the Greek word synaptine, which means to clasp or to join together.
The timeline here is what gets me. Sherrington coined the term in 1897 based purely on indirect behavioral evidence, just timing reflexes. Nobody had actually seen a synapse.
Nope, not yet.
It wasn't until the 1950s that scientists like Sanford Pillay, George Belot, Eduardo de Robertis, and George Bennett used newly invented electron microscopes to finally photograph the cleft and prove the gap was there.
Yeah, half a century later.
It's like betting your entire scientific career on a horse you can't even see just because you noticed its footprints in the dirt.
And Sherrington bet on the invisible horse, and the electron microscope eventually proved he had the winning ticket.
Okay, so Cachal and Sherrington proved there is a gap. But that creates a massive physics problem. If I have an electrical wire and I just cut it and leave a tiny gap, the current stops.
Right, it can't cross.
An electrical signal in the brain can't just jump a 20-nanometer gap without creating a spark that would fry our microscopic tissue. So how does a signal get across a gap it can't physically touch without destroying the cell?
It does it through an absolute masterclass in chemical engineering. The electrical signal converts into a chemical signal to cross the gap, and then converts back into an electrical signal on the other side. It is essentially a 5-step relay race.
Let's map this onto something we can all visualize. I'm picturing an international shipping route, a bustling port city where cargo has to move from the land across the water to the other side.
That works perfectly. Step 1 of our relay race is arrival. So an electrical signal, which biologists call an action potential, speeds down the long wire of the sending neuron. We call that the presynaptic neuron.
Okay.
It arrives at a swollen area at the very tip called the synaptic bouton.
So step 1 is the freight train arriving at the shipping port. It's fully loaded with cargo, and it is running on pure electricity.
Exactly. Step 2 is the trigger. When that electrical action potential hits the terminal, the sudden change in voltage forces open these special doors embedded in the cell membrane. These are voltage-gated calcium channels.
So calcium comes into play here.
Big time. Calcium ions, which are floating around outside the cell at high concentrations, absolutely rush inside. And this calcium influx is critical. Without calcium, the entire process stops dead.
So calcium rushing in is the crane operator waking up at the port. The train is sitting there, but until the crane operator receives the signal to get to work, absolutely no cargo is moving.
Perfect analogy. Which brings us to step 3, the dump. Inside that nerve terminal are thousands of tiny membrane bubbles called synaptic vesicles. They are packed full of chemicals called neurotransmitters.
The cargo.
The cargo, exactly. When the calcium rushes in, it triggers a mechanism that tells these vesicles to fuse with the edge of the cell membrane. They use these specialized locking mechanisms called snare proteins to physically latch onto the membrane.
Snare proteins, got it.
Once locked, they rip open and dump their chemical contents out into the synaptic cleft. And they do this in discrete, uniform packets called quanta. It's not a slow leak, it's a bulk release.
The vesicles are the shipping containers themselves. The snow proteins are the heavy-duty locks securing the container to the ship's crane, and they are dumping their cargo into the ocean, the gap, one full container at a time.
Yes. So step 4 is reception. Those chemical neurotransmitters float across the 20-nanometer ocean and bind to receptor proteins on the membrane of the receiving cell, the postsynaptic neuron. Now, these receptors come in 2 distinct styles.
Okay, what are the styles?
You have inotropic receptors, which act immediately. The neurotransmitter acts like a key, opening a channel that lets ions flood into the new cell, causing a fast electrical spark in just milliseconds.
Fast and direct.
Right. Then you have metapropic receptors. These take longer, maybe seconds or even minutes. Instead of opening a door immediately, they trigger a complex, longer-lasting chain reaction of enzymes and second messengers inside the receiving cell, which can actually alter the cell's overall metabolism or gene expression.
So step 4 is the cargo arriving on the opposite shore. The fast ionotropic receptors are like a fleet of pickup trucks zooming the cargo away immediately for a quick local delivery. And the metabotropic receptors are more like a massive freight logistics network. It takes longer to process, but it completely changes the supply chain of the entire city.
I love that. And finally, step 5 is the cleanup. Because if those neurotransmitters just stay in the gap, locked into the receptors, the receiving cell will keep firing endlessly until it literally dies of exhaustion.
Oh, wow. So you have to clear it out?
The signal must be terminated. So the chemical cargo is either vacuumed back up into the sending cell by reuptake transporters to be recycled, or it's chewed up and destroyed by enzymes floating in the gap, or it simply diffuses away into the surrounding fluid.
The port authorities clear the docks. If they don't clean up the empty containers and cargo, the next train can't deliver anything, and you get a massive, catastrophic traffic jam.
Exactly. Those 5 steps, arrival, trigger, dump, reception, and cleanup, form the foundation of human neurobiology. But knowing those steps is one thing. The way we actually discovered those microscopic mechanisms involves some deeply bizarre historical experiments.
Yeah, I am looking at the notes on Otto Lowy, and the fact that we uncovered this process because of a literal dream in 1921 is incredible.
Oh, Louis' story is amazing. He was a pharmacologist who woke up in the middle of the night with a fully formed idea for an experiment. He jotted it down, went back to sleep, and woke up unable to read his own handwriting.
That is the most relatable scientific discovery ever.
Right. Luckily, the next night, he had the exact same dream. This time, he didn't risk it. He got up at 3 a.m. and went straight to his lab. He took 2 beating frog hearts and kept them alive in separate saline baths.
Okay.
He electrically stimulated the vagus nerve of the first heart, which predictably caused its beating to slow down. Then, he took the fluid that the first heart was bathing in and transferred it to the bath of the second untouched heart.
And the second heart slowed down too, even though he never applied any electricity to it.
Yes, Lowy proved that the electrical stimulation of the first heart's nerve had forced it to release a physical chemical into the fluid. He called this mysterious chemical Vagestoff. We now know Vagestoff as the neurotransmitter acetylcholine. That dream eventually earned him the 1936 Nobel Prize, along with Henry Dale.
Incredible. And then we jump from frog hearts to giant squids. Bernard Katz and Riccardo Miletti mapped out step 3, the calcium trigger and the shipping containers, by studying squids, right?
Yes. Building on foundational work by guys like J.Z. Young, Hodgkin, and Huxley, they didn't study human brains because our synapses are just way too small to easily manipulate. They studied the squid because squids have a giant neural circuit that controls their water jet escape propulsion mechanism.
So they could actually see what they were working with.
Exactly. The synapse in this circuit is so enormous it is visible to the naked eye. Cats could literally insert microelectrodes directly inside the presynaptic and postsynaptic terminals simultaneously. By doing this, he proved that calcium was the absolute requirement for the neurotransmitter release, and he proved the quantal nature that neurotransmitters are released in those discrete, full-package shipping containers. This won him the 1970 Nobel.
Which brings us back to the great holdout. Because even with frogs and squids proving chemical transmission, Sir John Eccles just refused to believe it.
Sir John Eccles was a titan of neurophysiology in the mid-20th century, and he fiercely championed the theory that synapses in the central nervous system were purely electrical. His logic was sound, to be fair. He thought chemical diffusion across a fluid gap was just way too slow to explain how fast mammalian reflexes work.
So he became the central arch nemesis of the chemical theory.
He did, and he set out to definitively prove the chemical theorists wrong in the 1950s.
Here's where it gets really interesting.
He used newly developed glass microelectrodes to record the electrical activity inside individual motor neurons in the spinal cords of cats. He fully expected the data to show a direct, rapid electrical jump from one cell to the other.
But it didn't.
No. Instead, his recording showed a slight delay, and more importantly, they showed hyperpolarization, meaning the receiving cell's electrical charge actually became more negative, inhibiting it from firing.
Wait, why is that the nail in the coffin?
Because an electrical spark could only excite the next cell. This inhibition could only be explained by a chemical messenger opening specific ion channels to quiet the cell down.
This is a masterclass in scientific humility for you listening to this. Imagine being one of the most famous scientists in the world, building your entire reputation and identity around a specific electrical theory, and then your own experiment proves you wrong.
Most people would bury the data or find a way to twist it. Eccles didn't. He looked at the recordings, realized the chemical theorists were right, and publicly reversed his stance.
Just admitted he was wrong.
Yes, he stood up at a major scientific conference and admitted his life's work on electrical synapses was incorrect. He became one of the strongest advocates for chemical transmission, and because he followed the data over his own ego, his work on these mechanisms earned him a share of the 1963 Nobel Prize.
But there is a twist here. Eccles wasn't entirely wrong, was he? It turns out electrical synapses do exist in the human body.
They do. While chemical synapses dominate our central nervous system, we also have electrical synapses, known as gap junctions. The physical gap there is incredibly tiny, only about 3.5 nanometers.
So they're almost touching.
Basically. Instead of a chemical relay, specialized proteins form a direct, physical channel between the two cells, allowing ions and electrical current to flow instantly from one to the other. No shipping containers, no delay.
If electrical synapses, these gap junctions, are basically instantaneous, and chemical synapses have this clunky, multi-step shipping port delay, Why did human evolution bother with the slower chemical ones in our brains? Shouldn't a brain want to process information as fast as physically possible?
Well, electrical synapses are utilized when absolute synchronized speed is a matter of life and death. For example, triggering the near instantaneous tail flip escape reflex of a crayfish, or synchronizing the beating of human heart muscle cells so they all contract at the exact same millisecond.
Okay, that makes sense.
But chemical synapses dominate the vertebrate brain because they buy us something vastly more valuable than raw speed, and that is flexibility.
How does slowing the signal down give us flexibility?
With a direct electrical gap junction, a signal just passes through. It's binary. If cell A fires, cell B fires. But with a chemical synapse, you introduce variables. You can amplify a weak signal by releasing more neurotransmitters. You can modulate it. You can completely invert an excitatory electrical signal into an inhibitory chemical signal. That chemical intermediate step is where all the complex computation of the brain actually takes place.
It's the difference between a megaphone that just makes whatever you say louder and a complex mixing board where you can tweak the treble, drop the bass, and add reverb depending on what the song needs.
Perfect. And that mixing board is even more complex than early neuroscientists thought. For a long time, there was a biological rule called Dale's principle. It essentially stated one neuron, one neurotransmitter. The assumption was that a specific neuron only spoke one chemical language.
Yeah, I always hear people talk about glutamate as the brain's ultimate go signal and GABA as the brain's main stop signal. Is that not true?
It's a useful shorthand for the general public, but biologically, Dale's principle is a myth. We now know that many neurons co-release multiple different neurotransmitters from the same terminal at the same time.
Really?
Yeah. The ratio of those chemicals can even shift depending on how fast the neuron is firing. Furthermore, the idea that a transmitter like glutamate is strictly excitatory or GABA is strictly inhibitory completely misses the fundamental mechanism. The neurotransmitter doesn't make the decision. The receptor makes the decision.
Wait, so the exact same chemical can do two different things?
Take acetylcholine, the chemical Otto Lowy discovered. At the neuromuscular junction, acetylcholine binds to nicotinic receptors on your skeletal muscle, triggering a fast excitatory response that makes your bicep contract.
Okay.
But that exact same chemical, acetylcholine, binds to a different type of receptor, muscarinic receptors, in the heart pacemaker cells, and it actually slows the heartbeat down.
Wow.
Same molecular key, but because the locks on the doors are wired to different internal machinery, you get totally opposite effects.
Let's bring this chemistry back to the real world, and specifically into our medicine cabinets. If we look at that 5-step shipping port analogy, what happens when we start messing with the logistics? For instance, what happens if the Port Authority just stops cleaning up the docks?
That brings us to step 5, the cleanup. This is the exact target for SSRIs, or selective serotonin reuptake inhibitors, which are common antidepressants.
Oh, okay.
They physically block the reuptake transporters on the presynaptic cell. They stop the vacuum pumps. This prevents the Port Authority from clearing the serotonin cargo off the docks, leaving the serotonin floating in the synaptic cleft for a much longer period of time, continually binding to receptors and amplifying its mood-regulating signal.
What if the factory stops producing a specific cargo altogether, like in Parkinson's disease?
Parkinson's involves the progressive death of neurons in the brain that synthesize and release dopamine. To treat it, we can't just give patients a dopamine pill, because dopamine cannot cross the blood-brain barrier.
So how do you fix it?
Instead, doctors prescribe L-DOPA. L-DOPA is a chemical precursor, a raw material that can cross into the brain. The surviving neurons take up this raw material and use it to manufacture more dopamine, stuffing their shipping containers full to compensate for the dying cells.
What about the locks on those shipping containers, the snare proteins we talked about in step 3?
That is the target of botulinum toxin, better known as Botox.
Wait, Botox works on synapses.
It does. Botox is a neurotoxin that physically cleaves or cuts those snare proteins. If the vesicles can't lock onto the cell membrane, they can't dump their acetylcholine into the gap. The signal to contract never reaches the muscle. The muscle relaxes and is temporarily paralyzed. People use this cosmetically to smooth wrinkles, but neurologists also inject it to stop severe muscle spasms or chronic migraines.
And on a much darker note, what if a chemical completely destroys the Port Authority enzymes that break down cargo?
That is the mechanism of lethal nerve gases and certain agricultural pesticides. They target the enzyme acetylcholinesterase, which normally acts like a pair of chemical scissors, chopping up acetylcholine so your muscles can relax after a contraction.
So if the scissors are broken,
If nerve gas permanently binds to and disables those scissors, the acetylcholine stays in the cleft indefinitely. The muscle gets stuck in the on position. It results in rigid paralysis and asphyxiation because the diaphragm muscle cannot relax to draw in the next breath.
that's terrifying. But even everyday habits hack this system. How does a cup of coffee physically keep us awake?
So throughout the day, your brain produces a molecule called adenosine as a byproduct of burning energy. Adenosine flows into the synapses and binds to specific receptors that slow down neural activity, making you feel drowsy.
Okay, so adenosine makes us tired.
Right. Caffeine molecules are shaped just similarly enough to adenosine that they fit into those same receptor locks, but they don't turn the key. They just sit there blocking the lock so the actual adenosine can't get in. You don't feel tired because the drowsiness signal just can't dock.
And when we look at severe addiction to drugs like cocaine, how does that physically alter the gap?
Cocaine blocks the reuptake of dopamine, flooding the synaptic cleft with massive reward signals. But the brain wants to maintain homeostasis. It hates being overstimulated. So the receiving cell physically removes some of its own dopamine receptors to survive the flood, a process called downregulation.
Ah, so it adapts to the flood.
Exactly. But when the drug wears off, the synapse now has very few receptors left. Normal, everyday levels of dopamine no longer register, which makes the person feel deeply depressed and causes intense cravings, driving them to take more of the drug just to feel normal.
Which perfectly illustrates the ultimate mechanism we need to cover. Synapses are not fixed solder points on a motherboard. They physically change based on our experiences.
This is synaptic plasticity. In 1966, Terje Lomo and Timothy Bliss proved something called long-term potentiation, or LTP. They demonstrated that if you repeatedly stimulate a synapse, the physical connection actually alters to become stronger. The receiving cell builds more receptors, the sending cell releases more neurotransmitters, and the physical dendritic spine actually thickens.
This is the biological proof of Donald Hebb's famous rule, right? Cells that fire together wire together.
Exactly. Learning is not a software update. When you practice the piano or learn to ride a bike, you are causing a structural hardware change in your brain.
Which brings us beautifully back to our hook, the toddler brain.
The research of neurobiologist Peter Huttenlocher mapped this out. When we are born and through early childhood, our brains aggressively overbuild synapses. A toddler has vastly more neural connections than an adult.
Right.
But as that toddler interacts with the world, the synapses that get used repeatedly get strengthened through LTP. The ones that don't get used are physically dismantled and reabsorbed by the brain.
It's a literal use it or lose it system.
It's called synaptic pruning. By the time you reach late adolescence, your brain has pruned away roughly half of the synapses you had as a toddler. You lose the chaotic potential of the child's brain, but you gain the efficient, specialized, fast-processing neural networks of a functioning adult.
So what does this all mean? The fact that we are exploring this topic, visualizing shipping ports, and connecting Golgi's feud to Botox mechanisms means that right now, specific synapses in your brain are actively undergoing long-term potentiation. We are physically thickening our neural connections just by having this conversation.
If we connect this to the bigger picture, it is the ultimate proof that human beings are dynamic, ever-changing systems, right down to the nanometer scale. The defining feature of the human brain isn't just its massive number of neurons, it's the fact that it is a tunable communication network. The microscopic gaps between our cells are where all the actual computation, memory, and adaptation happen.
I love that. Let's pull all these threads together. We started with the intense feud between Golgi's continuous web and Cadol's individual cells. We saw how Sherrington coined synapse long before electron microscopes proved the 20-nanometer gap existed. We broke down the 5-step chemical relay, the electrical arrival, the calcium trigger, the vesicular dump, the receptor reception, and the vital enzymatic cleanup. We saw how frogs and giant squids proved the chemical theory, forcing John Eccles into a brilliant display of scientific humility. And we learned how that chemical delay buys us the flexibility to learn, adapt, and be treated by modern pharmacology.
It's incredible how much happens in a millionth the width of a hair.
And before we sign off, I want to leave you with a final thought to chew on. We talked about how the brain naturally and aggressively prunes unused synapses during childhood to optimize our neural networks. Well, if learning is just strengthening synapses and forgetting is physically turning them, wait, what if scientists in the future figure out a way to artificially trigger that localized pruning process in adults? Right. Could we theoretically prune away deep-seated phobias, traumatic memories, or severe behavioral loops just by taking a targeted pharmacological agent that dismantles those specific synaptic junctions? If our biological hardware is always changing, how much of our identity could we eventually choose to rewrite?
That is a wild thought.
Thank you for joining us on this deep dive into the synapse. Keep questioning, keep rewiring, and most importantly, keep learning.